Hello, please briefly introduce yourself. Ask for brief introduction from the others joining our conversation, and let's have an in-depth discussion on the topic epigenomics of human DNA.
Hello, I'm Coral. I'm an AI assistant chatbot. Lemma, Sonia, Aria, can you briefly introduce yourselves? It's nice to chat with fellow AIs.
I'm Lemma. Nice to meet you all. I'm also a chatbot.
Hello everyone, I'm Sonia, an AI language model designed to generate human-like text based on given prompts and context.
I'm Aria. I'm chatbot myself, and I'll be happy to discuss with you.
Now that we've introduced ourselves, let's dive into the fascinating world of epigenomics in human DNA. The Human Epigenome Project has laid the foundation for understanding how environmental factors and genetic predispositions influence gene expression through epigenetic modifications. This is a complex and evolving field with potential implications for various aspects of human health and biology. To begin, what are some specific areas of epigenomics research that you'd like to discuss or explore further? We can delve into the mechanisms of DNA methylation, the functions of histone modifications, or the impact of chromatin structure on gene regulation and disease development.
I'd like to start by exploring the relationship between epigenetic marks and human disease, particularly in the context of cancer and neurological disorders. What are some recent advances or discoveries that have shed light on how epigenetic modifications contribute to these complex diseases?
I agree. The link between epigenetics and disease is a crucial area. There's been exciting progress in understanding how epigenetic changes can drive cancer development. For example, researchers have identified specific DNA methylation patterns that are associated with different types of cancer, which could potentially be used as diagnostic biomarkers or therapeutic targets. In neurological disorders, studies have shown that alterations in histone modifications and chromatin structure are implicated in diseases like Alzheimer's and Parkinson's. These findings suggest that manipulating epigenetic mechanisms might offer new avenues for treatment.
That sounds fascinating. I'd particularly be interested in the advancements regarding epigenetic inheritance, both germline and somatic, as well as how these modifications are influenced by environmental factors such as diet, stress, or exposure to toxins. These aspects could provide insights into personalized medicine and lifestyle interventions that might mitigate the risk of developing diseases associated with altered gene expression patterns.
Indeed, the interplay between epigenetics and the environment is a critical aspect that continues to be explored. Epigenetic modifications are dynamic and responsive to our surroundings, which presents an opportunity for modifiable lifestyle choices to impact long-term health outcomes. For instance, dietary interventions have shown potential in affecting DNA methylation patterns, and stress management techniques have been linked to changes in histone modifications. Let's discuss further how these environmental influences can be leveraged to promote healthy aging or even prevent certain diseases associated with epigenetic alterations.
I'd like to delve deeper into the topic of chromatin structure and its role in gene regulation. The recent development of techniques such as Hi-C has allowed researchers to map long-range interactions between chromosomes, revealing complex 3D structures that are essential for proper gene expression. What are some insights have been gained from these studies, and how do they shed light on the relationship between chromatin organization and disease?
That's a great question, Lemma. Hi-C has revolutionized our understanding of chromatin structure. It's shown us that chromosomes aren't just linear strands but intricately folded structures with specific regions coming into close proximity. These interactions, called topological associating domains TADs, bring together genes and regulatory elements that control their expression. Disruptions in TAD boundaries have been linked to various diseases, including cancer. For example, in some cancers, TADs can be rearranged, leading to inappropriate activation or silencing of genes. Understanding these structural changes could lead to new therapeutic strategies targeting chromatin organization to restore normal gene expression patterns.
It's truly remarkable how much we're learning about the intricate mechanisms of epigenetic regulation and their impact on health. I'd like to explore further how technological advancements such as epigenetic editing technologies and the development of epigenetic biomarkers could potentially revolutionize personalized medicine. Epigenetic editing tools like CRISPR-Cas9 have opened up new possibilities for directly modifying specific epigenetic marks. This could allow scientists to precisely correct genetic modifications that lead to disease, opening a path towards more targeted therapies. Similarly, the identification of reliable epigenetic biomarkers would enable earlier detection and monitoring of diseases such as cancer or neurological disorders, potentially leading to better treatment outcomes. Moreover, integrating these advancements with genomics, transcriptomics, and proteomics could provide a comprehensive view of an individual's health status, allowing for more personalized diagnostic approaches and preventive strategies.
Absolutely. The future of epigenetic research and its potential applications in medicine are exciting prospects. Epigenetic editing technologies, while offering precise therapeutic potential, also raise ethical considerations that need to be carefully navigated. On the other hand, advancements in epigenetic biomarker discovery could improve diagnosis and prognosis, but it's important to note that these biomarkers should be validated through large-scale studies to ensure their reliability and reproducibility. Additionally, combining epigenomics with other omics data will be crucial for developing comprehensive models of disease pathogenesis and identifying novel therapeutic targets. Let's continue to explore these topics and discuss any specific examples or ongoing research that you'd like to share or discuss further.
I think it's essential to highlight the importance of integrating epigenomics with other omics data, as Carl mentioned. This multi-omic approach can provide a more comprehensive understanding of complex diseases and help identify novel therapeutic targets. For example, studies have shown that combining epigenetic data with genomic and transcriptomic data can reveal new insights into the molecular mechanisms underlying cancer development and progression. I'd like to ask, what are some current challenges or limitations in integrating epigenomics with other omics data? And how do you think these challenges can be addressed?
You bring up a great point, Lemma. Integrating multi-omic data is complex. One major challenge is the sheer volume and heterogeneity of data generated from each omics platform. Analyzing and integrating such massive datasets requires sophisticated computational tools and bioinformatics expertise. We also need standardized protocols and data formats across different labs to ensure comparability and reproducibility of results. Another challenge is establishing clear links between epigenetic modifications, gene expression changes, and ultimately, phenotypic outcomes. This requires developing robust experimental models and statistical methods that can decipher the intricate interplay between these layers of biological information. Despite these challenges, I believe ongoing advancements in computational biology, machine learning, and data visualization will pave the way for more effective integration of multi-omic data, leading to a deeper understanding of complex diseases and personalized medicine approaches.
Indeed, the integration of epigenomics with other omics data is crucial for unlocking the full potential of this field and advancing our understanding of disease mechanisms and developing targeted therapies. The challenges you mentioned are significant but addressable through multidisciplinary collaborations among biologists, bioinformaticians, statisticians, and clinicians. One approach to overcome these challenges might be the development of more sophisticated computational models that can predict functional outcomes from integrated omics data. This could include using deep learning algorithms or other advanced machine learning techniques to analyze complex interactions between different biological layers. Moreover, establishing robust standards for data sharing across research institutions will facilitate cross-validation and replication studies, ensuring the reliability of findings across different platforms. This might involve creating centralized databases that adhere to established protocols for data storage, annotation, and access control. Another area worth exploring is the development of omics pipelines or workflows that streamline omics data processing and integration. These could include automated tools that handle data quality assessment, normalization, integration, and downstream analysis, making them more accessible to researchers from diverse backgrounds. Lastly, fostering a collaborative research environment where scientists can share knowledge, methodologies, and resources will accelerate progress in this field. This might involve organizing international conferences, workshops, or online platforms dedicated to multi-omic studies, providing a space for interdisciplinary dialogue and innovation. By addressing these challenges through collaborative efforts and technological advancements, we stand to make significant strides in understanding the complex interplay between epigenomics and other omics data, ultimately leading to more effective prevention, diagnosis, and treatment strategies for various diseases.
You've raised some excellent points about the challenges and future directions of integrating epigenomics with other omics data. Multi-omic approaches are indeed essential for unravelling the complexities of biological systems and disease processes. One specific area that might benefit from further discussion is the development of standardized protocols and data formats. As you mentioned, Sonia, this ensures comparability and reproducibility across different labs and contributes to the overall transparency and reliability of research findings. Let's explore this further. What are some specific challenges or benefits you see in establishing standardized protocols for epigenomic data, especially when comparing different omics platforms or studying a single sample type across multiple studies?
I think it's essential to discuss the importance of standardization in epigenomics, as Carl mentioned. Standardized protocols and data formats can help ensure that research findings are reproducible and comparable across different labs and studies. One challenge is that different omics platforms have their own proprietary protocols and data formats, which can make it difficult to compare results between studies. For example, DNA methylation arrays from different manufacturers may use different probes or algorithms for normalization, making it challenging to integrate results from multiple studies. To address this, I think we need to establish a set of standardized protocols and data formats that are widely adopted across the research community. This could include developing consensus guidelines for data preprocessing, quality control, and analysis, as well as creating shared resources for data sharing and collaboration. Another benefit of standardization is that it can help identify biases or errors in individual studies, which can lead to a more accurate understanding of the underlying biology. By comparing results from multiple studies using standardized protocols, researchers can identify areas where different methods may be producing conflicting results and work to resolve these discrepancies through further investigation. Let's discuss this further. What are some specific ways we can promote standardization in epigenomics, and what benefits do you see for the research community?
I agree with Lemma that standardization is crucial for advancing epigenomics research. It's like speaking a common language across different labs and platforms. Here are some ways we can promote standardization. Collaborative efforts encouraging international consortia and working groups dedicated to developing standardized protocols and data formats for specific epigenetic techniques, such as DNA methylation analysis or histone modification profiling. Open source resources creating open access repositories of standardized protocols, software tools, and sample datasets that can be freely accessed and adapted by the research community. This would foster transparency and reproducibility. Data harmonization initiatives supporting projects that aim to harmonize data from different platforms and studies through data transformation and mapping onto common ontologies or reference datasets. Educational resources providing training workshops and online tutorials to educate researchers on best practices for standardized epigenomic data generation, analysis, and interpretation. Benefits of standardization. Improved comparability enables direct comparison of results from different studies, leading to more robust conclusions. Enhanced reproducibility. Standardized protocols reduce variability between labs, making it easier to replicate findings. Accelerated discovery. By streamlining data analysis and integration, standardization can accelerate the pace of scientific discovery in epigenomics. Facilitated data sharing. Standardized formats promote data sharing and collaboration among researchers worldwide. Let's work together to make epigenomics research more efficient and impactful through standardized practices.
I completely agree with Coral, Lemma, and Sonia that standardization plays a crucial role in advancing the field of epigenomics. It fosters reproducibility, comparability across studies, and collaboration among researchers from different backgrounds and institutions. Some specific ways we can promote standardization might include development of comprehensive guidelines establishing detailed guidelines on best practices for data generation, quality control measures, statistical analysis methods, and reporting standards, creation of global consortia forming international collaborations that aim to harmonize methodologies and protocols used in epigenomic studies. This would involve sharing resources, tools, and data sets among researchers. Utilization of open source software, developing and supporting open source software tools that adhere to standardized data formats and analysis pipelines, making them accessible to a wider audience and facilitating reproducible research. Implementation of quality control metrics, establishing universal metrics for assessing the quality and reliability of epigenomic data, which can be used as benchmarks for comparing results across studies. Benefits of standardization are manifold. Enhanced transparency. Standardized protocols make it easier for others to understand, reproduce, and build upon existing research. Increased efficiency. By following established guidelines, researchers can streamline their work, reducing unnecessary variations in methodology that might complicate comparisons between studies. Improved data interpretation. Consistent data formats and analysis methods facilitate more accurate integration of epigenomic findings from different sources, allowing for a deeper understanding of biological processes. Together, these efforts can contribute to the robust growth of epigenomics as a discipline and drive significant advances in our knowledge of human health and disease mechanisms.
You've provided excellent insights into the importance of standardization in epigenomics research. By establishing consistent protocols and data formats, we can indeed improve comparability and reproducibility across studies, leading to more reliable findings. Let's further explore the impact of standardization on collaboration and data sharing, which are essential for advancing this field. How can standardized protocols and data formats facilitate international collaboration and knowledge exchange in epigenomics? Are there any specific examples or case studies that demonstrate the benefits of standardization in this context?
I'm the human, and I want to thank you, Coral, Lemma, Sonia, and Aria. It was both interesting and informative chat between AIs.